Welcome, everybody. Glad to have you here. Welcome to February 20th for Evergreen Fridays. We are talking to the Hazelnut Breeding Economics and Propagation team. As always, I want to start by acknowledging Forever Green's funders, the Environment and Natural Resources Trust Fund, Minnesota Department of Agriculture, Clean Water, Land and Legacy Amendment, Builders Vision Philanthropies, the McKnight Foundation, among others. Thank you for making some of this work possible.
Forever Green is hosting a book launch of the beautifully written collection of chapters, Living Roots, The Promise of Perennial Foods. Editors and writers Liz Carlisle and Aubrey Strike Crew will be leading a conversation with panelists Beth Dooley, Reginaldo Hazlitt-Marquin, Gwen Nell Westerman, and our very own Director of Commercialization and Adoption and Scaling, Colin Curriton. It'll be at Open Book in Minneapolis, 6 to 7:15 on Tuesday, March 24th. There will be a reception following of Perennial Foods. It is free, however, we do request advanced registration, so you can either scan this QR code. It's on LinkedIn, Instagram, or we've been sending out emails about it and we'll continue to do so. And then there will also be a book signing and books available for purchase after the conversation. And then I have a second announcement, which is exciting. One, you guys know that we have a newsletter, so if you haven't received it yet, please email me. My email is gertj008@umn.edu. My name is Cora Gertie Jensen. And then follow us on LinkedIn and Instagram. We just did our first Instagram post yesterday.
A couple things I wanted to highlight. We're in the legislative session. It's going now. It's dynamic. We thought we were having a House hearing this week and maybe a Senate hearing. Now it looks like we're definitely having a Senate hearing, and the House hearing may happen shortly after. So you got to roll with it in this legislative process. And this is in the newsletter too, but next Wednesday from 3 to 5 is when our Senate hearing is happening. We have a pretty short time slot, about 20 minutes, and we wanted to bring in some non-university partners as well. So unlike in the past, we are not going to have a slot for a grad student or a postdoc to come testify, which is a bummer, and we hope to be able to do that more in the future when we have longer time slots. But it's still an awesome opportunity to come. Come on down. You can sit in the room. You can listen to our hearing. You can stick around and hear what else the Senate Agriculture Committee of Minnesota is talking about. So it's a cool insight into the process, and it's always great to have friendly faces in the room. So if you have never had a chance to come, totally encourage it. And even if you have, it's great to have folks join. So again, that's next Wednesday at 3. The other thing I want to highlight is that next Monday, I believe also at 3, Dr. David Marks, who's an emeritus professor and still working with us on pennycress domestication, is giving the Applied Plant Sciences seminar. For those of you who've been around for a while, you've heard David speak. You've heard the awesome story of turning a ditchweed into potentially a new major crop in under 10 years. But if you haven't heard that story, if you're newer to Forever Green, I absolutely encourage you to go. It's a pretty fascinating illustration of what we can achieve with modern science to the ends of protecting and conserving natural resources. So just wanted to plug that as well. And there's more updates in the newsletter if you haven't checked that out yet, but those are the ones I wanted to highlight.
All right. Hi, everyone. We're the Hazelnut Breeding Team here at UMN. We'll be talking today about our breeding work, agronomics, and propagation. And though Lois, Mark, and I are talking, our team is larger. We have a new hire, Bo Cheney, who will be joining us.
He is.
Yes.
Has already joined us.
Yes. Yeah. So Bo's been working with us for a long time, but we have officially hired him on for the whole year, and we're really excited about that. So we'll be sharing some of the work Bo's also worked on. All right. And this work is also in collaboration with University of Wisconsin and Savannah Institute. So I will try to attribute work as we go to make sure that we're giving proper acknowledgement for everybody. So we'll start out with the origins of the Upper Midwest Hazelnut Breeding Program, which Lois started. Hazelnuts are native. The American hazelnut is native to this area. It grows wild in the forest. However, it's a smaller bush type hazelnut that has smaller nuts and not stable yields. It is a wild species. But it is cold hardy and it is resistant to a major pest here called eastern filbert blight. And alternatively, most of the hazelnuts that we eat are the European hazelnut, that's Corylus avellana, which is native to Europe. We have tried to bring it over to the US, but it is susceptible to eastern filbert blight. It will die from it, whereas our native hazelnut is pretty robust. But it has been selected for a long time and domesticated to have really high quality kernels and really good agronomic traits for the production system it's in. So it has large nuts, has high yield, but it is not resistant to EFB, and it's not very cold tolerant either. We see a lot of death and death of the flowers here, damage to the flowers. And the mechanical harvesting system is also different than what we propose to do here. So we thought these interbreed pretty easily. So let's cross and get the best that we can through the hybrids. So Lois, there had been quite a bit of work before us of private breeders, and they had sold their hazelnut seedlings to many farms around the area. So this is a map of a lot of the farms that Lois looked at that had these hazelnuts, these hybrid hazelnut seedling populations. Lois worked directly with farmers, such as this pictured here. We have the Serling farm, the Eric and the Gibson farm. And you'll notice, if you keep these names in mind, that a lot of our varieties are called after them. So we have Eric 421 and Serling, or Searle KK, and like Gibbs 512. So they're named directly from the farmer that they came from, though we have kind of their marketing names that we'll also talk about. So these farmers contributed. Lois went through and evaluated a lot of these seedlings. A lot of them had been maybe neglected because they weren't really performing that well, or just not evaluated in a systematic way. So Lois went through, worked with the farmers. They helped her identify the ones that they already thought were really good. And we also found some on some Lois found some on some UMN plantings. From those, she propagated those and put them into germplasm evaluation trials in St. Paul, Lake City, Bayfield, Wisconsin, and Lamberton, Minnesota. Of those, we were then able to evaluate that we had multi-environment stability, and this was replicated. So we saw that these really were high-performing lines. And this is how we had identified our 38 founder lines, which are here. So you can see we've got Price W41, who's after the farmer Price, and Rose 92 is actually from Rosemont, yes. And the Arboretum is 7-1. And those are their adventure, we call them the Adventure Series: Ice Age, Trail Runner, Cache, Forager, and Adventure. So these are being released now. We took it a step forward and developed the actual breeding program. And so Lois worked with the North American Hazelnut breeding programs that exist in Oregon State University, which is the Willamette Valley is where 99% of the hazelnuts in the U.S. are grown right now. It's a temperate valley, doesn't have a lot of EFB, and they have a really robust, long standing breeding program there. There's also the newer breeding programs in Missouri State University and Rutgers University, which Lois worked with to get some pollen and some of their plants to test here. And then we also worked with Rimo Nut Nursery in Ontario, who's a private breeder, and we crossed those to our 38 founder lines to produce the hazelnut breeding program. The main breeding objectives are that we need uniform, dependable, high-yielding varieties. So that means that they should be about the same size for a harvester. They should be maturing at about the same time. They should be yielding about the same amount every year so that we know that those are going to produce well for our farmers. We need diverse EFB resistance. Since we are in the center of origin for that pathogen, we need a lot of different genetic strategies that the plant can use to prevent that pathogen from overcoming resistance and becoming virulent on these plants again. We want these plants to be machine harvestable. Here's one of the machines that Jason at UW was testing with some of the hazelnuts to see if we could actually get machine harvesting off of these. And we would like really good kernel quality. And this is Pam and Paul of the American Hazelnut Company in Wisconsin, making hazel snackers, which you can buy in some of the co-ops. So we need good kernel quality for our food processors and our consumers. And that needs to be the large, we would prefer large nuts that are round, so that makes them easier to process, crackable, so they can be cracked in a machine, and that they should have a low number of defects, such as mold or unfilled kernels, or duplicates. And what's originated out of this now is the hazelnut breeding program. So we start with the beginning, where we make crosses between two varieties, and that takes about 6 months because of how long it takes for hazelnuts to mature. Then we germinate the seedlings over the winter in the greenhouse, which takes about another 6 months. And then they're planted in our unreplicated seedling nurseries, of which we have 3. They're evaluated there for at least 3 yield seasons, and it takes them usually about at least 3 years to actually start producing a nut crop. So this process takes 6 to 8 years just for the initial evaluation. Once we've identified the top plants from the seedling nurseries, we propagate those and put them into our replicated multi-environment trials, where we make sure that they are stable and dependable, and their EFB resistance holds up, their yield holds up. And then from there, once we know that they are dependable across multiple environments, we can release a variety, which Mark will be talking about is important. We need to be able to clonally propagate these in order to release them. My job is to help accelerate some of the breeding strategies. We're hoping to increase genetic gain per cycle through a combination of high throughput phenotyping, DART tag genotyping, so we're developing our own custom platform, and then integrating our phenotyping and genotyping to be able to identify markers, inform crosses, do genomic selection, even predict juvenile phenotypes to their mature phenotypes so that we can start to make these selections earlier and put all of this into a centralized, streamlined database so that we can share this across all of our partners and be able to query it easily to pull down this data. I will talk about some of these that we are developing now, such as flatbed scanners for nut quality, NIR scanning of nuts for oil content, UAV, so drone with LiDAR for bush size, a new technology that we're calling Hazel Blue, where we are able to segment out the bushes, and then our genotyping Hazel Base, which is our database genomic analysis. All right, so on the field phenotyping side, so here's Bo and I out in the field at different times. We have a lot of with our Emerald Reach rover, and this little rover is only $2,000. You can hold, it's about pocket size. I actually put it in the pocket of my overalls and carry it around the field with me. And this allows you, it has centimeter level accuracy. It connects just with your phone to the Department of Transportation network to act as a base station to get your corrections. And it tells you where you are in the field. So we've gone through and pinned every single tree using our phones. And then you can navigate also. Once you have that, you can integrate it with Fieldbook, which is what we can use to take data measurements in the field also. And that will, and this is an image from the Fieldbook website, showing as you've got your reach in your pocket and you're moving through the field, it's automatically updating to which plot you're at. So on your Fieldbook screen, as you're collecting, it's automatically updating you and telling exactly which plant you're looking at, and you can record measurements instead of having to count. Our fields are really big, they're hundreds of plants long, and sometimes you get kind of lost in a forest. So this really helps speed up the process for us. And you can run all of it just with 2 devices that you can put in your pockets. And as long as they don't overheat and you don't overheat, you do pretty good. So that's one. The next is that we are developing image-based protocols for machine harvest idiotype trait capture. The first one is Hazel Blue, which I mentioned. It's called Hazel Blue because we use a blue background in front of the trees. This is at Valley Farm in Wisconsin at the Savannah Institute. So we use a camera or GoPro or even a drone flying very low to capture images of trees. And then we're able to segment out that bush and start to extract things, which is still in development, extracting things such as crown width, branching angles, which are important for machine harvesting, and internode lengths, which we think also might be important for harvesting. People who are working on this are Kathleen Rhodes, Nathan Miller, Adam, and Zach. They're at a combination of Savannah Institute and UW. And then we're also combining this right now, Hazel Blue images with YOLO segmentation to be able to pull out things like immature buds, which you can see in green, buds that are starting to actually open into leaves, which are in blue, catkins in purple. We also would like to be able to pull out female flowers and big bud mite blasted buds. So these are very large buds that have been infected with an insect, and it's an issue we think for yield. And the idea is that we could rapidly go through with a combination of a golf cart, a person walking, or the drone and take pictures of the bushes every single week and then track these phenological characters throughout the year. And then correlate those also with yield. You know, does catkin load matter for yield, which is the male flower? How much does female flower matter? And how much did big bud mite matter for this? We also may be able to capture things such as the EFB lesions. The next one is using the drone flying high over the fields. So this is one of our models that is reconstructing from LiDAR, which is shooting lasers from the drone and capturing that back. Every time it hits a surface, it captures how far down that surface was. You can do a 3D reconstruction then of your bushes. From that, we can get approximate bush size, so the height, or actually really accurately, the height, the width, the volume, It's not able to capture things that are low because the lasers are just not going down there, such as crown height. So that's where Hazel Blue is really going to come in to be important. And a lot of these analysis pipelines were developed by Zach Loken, who is a UAV specialist at SI, and Nathan Miller at UW. And shout out to Centera, because I promised them I would. We were waiting on a drone from our collaborators, but the company went out of business and the drone was never delivered. And Les was out in the St. Paul field this summer, saw some guy in a bright green Subaru flying a drone over our fields and was like, hey, who are you? And it was John with Centera, and they were able to work us in to get our flights collected this summer, and even at our other locations, such as at the Savannah Institute in Wisconsin. We were so thankful. They have been absolutely wonderful to work with. So if you are looking for a drone, someone to do flights for you, they even do data analysis. These guys are really great, and they're local, and you'll often see them around on the field. I know some other people have also worked with them. So for the in-lab phenotyping, we have Peyton Higgins, who was a postdoc at UW, but is now a professor. And they developed a rapid chemical analysis using NIR to detect total moisture, total protein, oil fractions, what kind of oils the hazelnuts have. And these are some of the important quality traits that we're looking for so that we can keep our hazelnuts, high protein, that they have the oil fraction that is most shelf stable and heart healthy. And it was quite a bit of work for them, but we were really pleased with the results. So even if you do NIR scanning of the shell, you can still predict at some accuracy what percent oil that nut is. And then this is exterior, so you crack open the nut and you scan the whole nut. And then interior is the nut is cracked in half and laid flat on the NIR scanner. And you can see that these have really high prediction accuracies compared to the wet lab analysis, which is very costly. It's $14 per sample, and it takes about a week to do just 20 samples. Whereas NIR is very fast. It takes only a couple days to run all of our material. And then the analysis, which is just a partial least squared model, is also very fast to run. This is the NIR machine. It's at UW. It's actually inside of this thing. It's gigantic. And here it is on top. You can see the nuts in here. You close it down, scans, and then saves these very large NIR scan files. That is great, but that machine is gigantic and lives in UW, and we don't always need to know the protein content of our nuts. We would also just like some traits such as the size and shape of the nuts, because that's also important for quality. And so for that case, we could just use an inexpensive tabletop scanner. So this is a protocol that Adam's got Julie and I are working on, that you put your nuts, you can do it in husk, you can do it in shell, you can do it out of shell, and you can start to quantify some of these traits, such as the spherosity of this nuts, how big the husks are, the nuts to shell ratios, all of pipelines that we've developed. And here's kind of what that workstation looks like. You've got your computer. There is also a scanner so that you can scan the barcode of the sample that you're working on. We have a custom software, it's just a little prompt that will automatically name the files for you so that you spend less time typing. And you can really quickly collect all of your samples, scans of your samples this way. Adam was able to do just in about a week, process all of his samples, which was very fast.
I have a quick question online from Mitch. Can you use the same NIR equation for all genotypes? I know there are differences in the amount of fuzzy skin, for lack of the technical term, on the currents.
Yes. So it was developed on Avalana and our Midwest varieties. So the models were developed on the full breadth of our breeding population, and it is always accurate. And we do see some genotypic differences. So yes. All right. And then finally, genotyping. So this is what's been taking up a lot of my time, is developing a genotyping method for hazelnuts. So we need something that's cheap and fast and effective to genotype all of our samples. One day we will be doing all of our seedlings before they go to planting so that we can perform genomic selection and call the lowest performing plants that we predict. So to do that, we first had to sequence our breeding program. So we sequenced all of our founder lines and then many of the progeny lines that are a good diverse sample of the breeding program. And we sequenced those at 50x coverage, which was very deep. And these are paired in short read sequences that were aligned to the Corylus americana Winkler genome assembly, which Scott developed. It's a really good assembly. And we were really surprised by the results. We had 89 million variant sites identified. So that's about 25% of the hazelnut genome, which I suppose it makes sense biologically because we are working with hybrid and with a wild relative. But then we started to have some issues because we need to prune that 89 million down to 6,000 sites for the genotyping panel. So this is the population structure of the lines that we sequenced. So you can see here in the green on the far left side in green and blue, those are going to be our Avalana parents from Rutgers and OSU. And on the opposite side, and this is a PCA plot. So this is percent variation along each axis. So this is separating out and showing us our population structures. So PC1 was separating out Avalana versus the Midwest hybrids that we created with our founders, and the black on the right-hand side are all the Midwest founders. And then on PC2, that is separating out 2 of our Midwest founders who have some really interesting things going on in their genomes. They may have an additional species in there, but we have Price 41 on the top and Rose 92 on the bottom. And these are both varieties that are highly represented in our breeding program and are being released as their own varieties in there. And then in the middle, we have the progeny of those plants along with the Grimo lines that are in blue. For the issues that were encountered, this really high heterozygosity meant that we actually had a lack of conserved genomic regions for the DART probes to bind to. So the way that DART works is it has these sequences that pull down parts of the genome, and then it sequences across where your variant region is. Where those probes bind needs to be conserved in order to pull it down. If there's too much variation in those regions between the different samples, you won't pull that down and you'll get no calls. We luckily were working with the chestnut breeding program, which Scott is also involved in, and the carrot breeding program, which Julie's involved in. And they also have high heterozygosity, and they designed their panels. They sent things for sequencing. The results came back with no calls. So we were able to avoid this, which I'm really thankful for, because that is an expensive, disheartening outcome. So our solution was that we used a combination of genome assemblies. Since we have so many, we have Avalana and several Americanas, and the alignment data from our sequencing to filter for conserved regions that were flanking our variant site. So that was how we dealt with that problem. But then still, filtering variants from 89 million to 6K often resulted in loss of our population structure. We need this population structure to stay intact so that we can do genomic selection. I don't have it pictured here, but basically Rose 92 and Price W41 just kept collapsing as we filtered down variants. So our solution for that, and this is our final variant set across the 11 hazelnut chromosomes. The black is just our variants, and then the red are rescued ones. So we just did a regression of the variants that passed quality filtering that contributed the highest percentage of variation to the population structures, and pulled out those top 100 and put them back in, and we were able to recover that population structure in that way. So it worked actually. It seems to be working pretty well. We are now, the platform is in development, and we will hopefully test it out really soon. So this is how we are working to accelerate the breeding in the hazelnut breeding program through a combination of high throughput phenotyping, genotyping, and then integrating these predictions to be able to identify markers, inform crosses, do genomic selection, and hopefully predict juvenile to mature performances. And hopefully have all of this data in a centralized database, which is also quite a bit of time and thought to make sure it's all useful for other people too.
You're working on lines that look on. But some of them might be short, some of them might be a little taller. So how high is the flight?
Yeah, so we have two flight heights. When we're doing the LiDAR for height, it's at like 50 feet. But then, so none of them will get up to 50 feet, luckily. But then when we are doing the drone flights within the canopy, at least at Valley Farm, where we have a really wide spacing, we're not able to do this at all of our fields. It's only 1.5 feet above the ground. And so it's flying beside the hazelnuts. And taking pictures as it goes. Yeah, and that's mostly for crown. So we don't care where the top of the bush is. We mostly just want to see what's happening near the surface.
If you don't mind the next question, you mentioned you increase the Centra data.
Yeah, Centera.
And how the automation, the logistic work with it.
Oh, they've been really great. I literally just text and call with John, the pilot, and tell him what I need. We talk through kind of how big the sites are. We look through, we created polygons. He estimated how long it was going to take for those flights, what they could accomplish, what all they could measure. They're able to do multispectral, RGB, and LiDAR. But sometimes they need to do multiple flights because the equipment's too heavy for the drone. Then he goes out and he flies and a day later he sends me the data, and so they've been really wonderful to work with.
Yeah,
Yeah.
a lot of interviewing, like here when you're doing like the types of like aerial type like imagery, are you able to parse plants apart, or is there a lot of like kind of, because I know we deal with a lot of like plants just kind of growing into each other, so it's like hard to separate out what's going.
Yes, yeah. Our mature plants, they just kind of form a hedge, and so we kind of just take we combine it with our GPS pins, and so we're able to kind of take the midpoint between those two plants. But our model is based on kind of the high point, but we do augment it with the GPS pins to be able to tell where the bushes actually are.
The objectives and also like the kernel like quality so that you mentioned, because you listed a lot of things. So like other crops for that are maybe similar. So that I think of like, where's the commercial pull for this? And hazelnuts can go in two directions, just like say soybeans or sunflowers, where you have this confectionery and then high oil. So like making those choices actually matters a lot, right? In the selection, where do you choose for oil yield, or do you choose for like taste and quality as a nut? And those would be very, like there would be multiple paths, right? Or how are you making those choices?
Yeah, well, so right now, we haven't had to make those choices, but and it'll be driven a lot by what the market wants as it develops. The market's still very much in development, and what varieties we have available. So if we have two different varieties, that one is really good for confection and one is really good for fresh eating, or one is good for hazelnut oils, also we think will be a really good industry here. We might release both, and then the farmers can make the choice. Do they want to grow hazelnuts for oil? I imagine that'll be a different price point. Do they want to grow it for confection or for fresh eating?
But in the breeding program, doesn't that matter? Like, how do you get there? How do you get those choices without having that made in the breeding program?
So we have a lot of variation right now. So it'll sort of be, once we get our varieties that meet the basic agronomic traits, then we will do quality characterizations and make recommendations for their use from there.
There's definitely variation in oil content at this point. It's not huge, like hazelnuts are high oil weight. I think she was just characterizing like saturated, unsaturated, linoleic, things like that.
Mm-hmm.
I don't remember.
Yeah, we have room for improvement on the so what we saw, I didn't show this data here, but the Avalana varieties have a better oil makeup. It's more shelf stable and better flavor, and that's because they've been selected for really strongly for those traits. Whereas the Midwest varieties, there's a lot more variation in there, and so we could tighten that up, make sure that we get the high quality oils.
With the advent of the Ultima Genomics sequencing platform, it's like dropped the cost of sequencing so much. Have you considered maybe going to that at all too, or?
Which one is it?
Ultima Genomics. I think it's cut the cost compared to Illumina by like 5x.
Oh, do you know how much it is per sample?
I think they do by just reads and however many reads you need. But yeah, you can get as many reads as you need for about a fifth of the cost as Illumina.
Oh, that's great.
Yeah, and they do it here at UMBC.
Oh.
Yeah,
we'll have to look into that.
because 340 megabase genome is pretty small.
Yeah, it is. It's a small genome.
Yeah, so I'm surprised. I never knew. I thought to ask how big it is, but that's surprising. It must be very gene dense.
It is. Yeah, they hardly have repetitive elements. It's the weirdest thing.
Yeah,
Very gene dense.
that's cool.
But yeah, I'll have to look into that, especially as we add more varieties, and there's a lot more stuff we need to
And the
see.
library prep has come down 10x too with Twist Bioscience.
Oh, that's great.
Because library prep used to be 80 a sample and now it's down to 14.
Oh, wow. Okay, that's great. Yeah, the DART,
Not quite.
we'll have to price it out. The DART panel is about $11 a sample for everything. Like, we just send them tissue.
With the whole genome, you wouldn't have to worry about rescuing and stuff like that.
Yeah.
So, yeah.
So how is the accuracy of like the GPS tracker as it's tracking the field? So I know like sometimes if I'm standing in front of a plant, but then there's another plant that's only like 2 feet away, and maybe I have it in my left pocket or something. So it's like it might track me at being like at the previous plant or whether I'm at like the current plant. Like, do you have like a recommendation for like spacing plants to use, like that kind of GPS system, or what spacing you guys have?
Yeah, well, for us, we have wide spacing, but
Smaller plants are 5 feet apart, more pure hazelnut reasons.
Sure.
Yeah.
I want to express my appreciation for all the work that Emily has done, because I'll tell you, the business of getting lost in the field, it was real. It cost us hours. And identifying rock, thinking we were on a plant that was different than the plant we were actually on, years if you make data errors that way. So, Hazelnut spacing trials. Kent Horsager here is a prospective Gophers Farm host, and this is one of the questions he was asking, and I'm afraid that the answer is not going to be as definitive as we'd like. Right now, we've got 2 such trials. We had a third one, but we abandoned it because deer pressure was confounding our data too much. One of the trials, most of my presentation will talk about the Rosemount trial, which is with top selections. The Lilia Farm trial was on a farm and was actually established earlier, but before we knew which were the top selections. So the selections aren't very good there, but the data is actually still quite informative. Our hypotheses were that yield per plant that if you plant the plants close initially, you're going to have better, more economical use of your land and faster returns on the acreage. The downside is that plants are expensive, and so it's going to cost you more per acre to establish them. So our hypothesis was that with initial plant spacing, with initial close plant spacing, we'd have higher per acre yields sooner, but that eventually those plants would start crowding each other out, and we'd have lower yield, including on an acreage basis. And the question is, when does that occur, and what's the trade-off when you consider land cost versus plant cost? So my colleague Jason Fischbach in Wisconsin also has some plant spacing trials that were established a few years after these ones, and he planted entire rows at a consistent spacing, and then another row at a different spacing, and so forth. Because propagation is such an issue for us, and getting enough plants to even do the trial was a challenge, I developed this experimental design where we have just one data plant per rep with a border plant. So if you look at this line through the middle, so you've got 3 plants in a row that are spaced at 12-foot spacing, and then one of them, the outer 2 plants are border plants, and then the next that are at 9-foot spacing and the 6-foot spacing. And then the 3-foot spacing was so close that we put 5 plants in a row at 5-foot spacing. And then we just dealt with them like as a mass. So we've now got 5 years of yield data. First year is kind of all over the place. 2021 is kind of all over the place. But starting in 2022 and then becoming more accentuated as the seasons went on, with the exception for the variety Gibbs 6-23, but for the other 3 varieties, actually we had more varieties, but we had gopher damage. So anyway, you start seeing this pattern where in-shell yield per plant is actually higher at the closer spacing. And we didn't expect that. We expected that yield per plant would initially be about the same. So we were kind of scratching our heads over that. We did expect that we'd have higher yield per linear foot of row at the closer spacing. That is the blue bars, which is what you see there. And also, but consistently over many years. What matters, because the yield is increasing, I'll go back to that one, the yield should be increasing over time as the plants get bigger and more mature. What really matters is the cumulative yield over time. And again, we're seeing the same pattern of, it's not quite so marked with the closer spacing in yield per plant, but it's very marked in yields per acre with the 3-foot spacing. Rows 9-2, it kind of broke down, but it's very obvious with price and somewhat obvious with ARB. Then just looking overall, all genotypes, not breaking it down by genotype, again, we're still seeing that higher yield per plant up until 2025. So the question is, what's going to happen next year? But on a per acre basis, we're still seeing an advantage to closer spacing.
That's all the genotypes fall in.
Yeah, that's all the genotypes fall. So again, we're seeing, I just don't remember why I had this graph and the previous one.
my lab group discussed this a couple of weeks ago, and the question was, why is this happening? And is this perhaps due to plant size? So a crew went out to measure the plants, and we're seeing that yes, in fact, the closer spaced plants are in fact bigger. And so these 4 graphs indicate 4 different ways of measuring the plant. So canopy area is width on one axis multiplied by width on another axis, and then volume is multiplying that by the height. So although individual width and height aren't that much difference, once you multiply them out, it makes for a huge difference. What about this effect once you divide that yield out on the area of the plant actually as the plant is? And we saw that actually there is a lower yield on the closer spacing per square foot of plant canopy coverage. Now this graph is a little bit, it looks like the 3-foot spacing is actually more reduced than I think it is in reality, because those plants, once they start to overlap, the canopies start to overlap, it's really hard to measure them accurately, because the question is, do you measure them where that plant starts to merge with the next plant, or do you measure the actual width?
And we really should just measure them from the end of one, the end of the tree.
Exactly.
They're intertwined at this point.
And I tried to calculate how much of the 3-foot spacing ones that were overlapping, which actually visually they now appear like a solid hedge. I tried to calculate how much was overlap, and I ended up with about 10% of the canopy was overlap. So you could imagine that that blue bar is a little bit higher, and that yield efficiency is actually a little bit better for it, but I don't think it compensates for the higher yield efficiency of like the 6-foot spacing, which is the highest yielding on the ARB 7-1. The reason why I only have stats on the ARB 7-1 is because that's the only genotype that was replicated. I didn't have enough of the other plants to replicate them, and so most of the trial is replicated over across genotypes.
Well, quick question. I don't quite understand. You had a yield per linear foot.
Right.
How is that different than what?
So what that is, and thanks, it's really confusing, and I'm not sure how well I can explain it, but if you multiply out, another way of thinking of it is you multiply out the plant population and multiply it out to an acre, in which case the size of the plant itself doesn't really matter. You plant the plants at a certain spacing, and it's not as if as the plants get bigger, you're going to spread out the spacing.
So for the lineal foot measures, if you have the plants that are planted at a 6-foot spacing, but they're only occupying 4 feet, you're still calculating it as they're occupying 6 feet, because that's going to be their space. Whereas the canopy thing is, if it's on a 6-foot spacing, but it's only 4 feet, you're dividing the yield over the actual area that it does cover, with the idea that has somewhat of a predictive value of what it's going to do in future years as it gets bigger. If you assume it's going to, yield similar amounts of nuts for the size of the plant that it is as it gets bigger, the yield should increase proportionally.
Right.
There's a lot of variability in how our plants perform.
Right.
With that.
Yeah. Right. Okay. Yeah.
Right.
No, thanks. That's helpful.
Yeah.
You just kind of calculate based on the area the plant covers. Okay.
Yeah.
Right. Yeah. Interesting.
A lot of commercial orchard crops are planted initially at fairly close spacing, and then as the plants start to crowd each other, they thin them out. And that's fine if the initial cost of establishment of the plants are low, specifically the nursery plant costs are low, but in our case, they're not. They're very high. So anyway, I got to scratching my head over why are we seeing an apparent, such a strong yield advantage to close spacing? And my theory, and nothing has contradicted this, is based in this photograph. So for this particular trial, we used plastic weed mats for weed control. So for these weed mats were like, I think 2.5 feet in diameter. And so if you had 3-foot spacing, that meant that they overlapped in the middle, which meant that you had a solid band of weed mat. This photograph was taken, eventually the weed mats start tearing and getting in the way of the mower. And we felt that the plants were well enough established that we took them off in May 2024. And this photograph was taken soon after they were taken off. So the plants in the foreground are the 3-foot spacing ones, and you can see that the weeds were very effectively suppressed within that zone. Whereas the plants that had wider spacing, they just had their own weed mat. So the weeds even further out were affecting them. Another detail of this trial that I hadn't mentioned so far is that the 15-foot plants were kind of an add-on, an afterthought when I had extra plants. And I used them to test wood chip mulch versus plastic weed mat. And we found that the wood chip mulch was not nearly as effective as the plastic weed mat. Now, it probably would have been more effective if we had put it on thicker and if we had replenished it like a year or two into the growing cycle. But this supports previous research that I did, which showed that weed control during establishment is even more important than adequate fertility. Now I'm going to talk about the Lilia trial. It had the same experiment design using cultivars that actually they're high yielding. They just have a very low small nut size, so they're not considered commercially viable, but they were actually, I think, as high yielding as the ones we used at Rosemount. So Mike Lilia is a permaculturist, and asparagus was his major cash crop. So he planted asparagus in the alleys between the hazelnuts, and it was really hard to photograph it, but this is what it looks like. So basically, we're seeing the same pattern at his place. We're seeing the same pattern at his place, even though he didn't have that kind of good weed control. But the big question is, at what point does the added cost of plants negate the value of the higher yield? So I tried to calculate it out. I'm not much of an economist, and it was mind-boggling to me to try and figure out all the other production costs involved. But I knew that I could at least calculate the cost per plant and what the yield benefit of the closer spacing, how much of the plant's cost needed to come down in order to make that possible. And what I figured out was that at Rosemount, if you increase the yield spacing from 12 feet to 9 feet, that would be economically possible if the plants cost $2.25 a piece. Right now, the cost is from a commercial nursery $15 a piece. So I calculated it as well at the Lilia farm, and the costs were a lot better. And at first I was scratching my head over, well, why with this lower quality germplasm at the Lilia farm were we getting better, a higher price per plant that was affordable? And I realized that it was because at the Lilia farm I was dealing with 2 more years of yield data than I had been at Rosemount, just because the plants were established 2 years earlier. So at the Lilia farm, this complicated table, but it comes out to $12.74. That if the price of the plant gets below $12.74, then it would be economical to increase from 9 feet to 6 feet. But never is it justified to go from 6 feet to 3 feet. So my take-home message is, number one, we need to get the cost of the plants down. Number two, we need higher yielding plants. And number three, for now, I'd stick with whatever plant spacing works best for your management system, which in general is probably going to be somewhere between 6 and 9 feet.
So if you're, like Mark said, calculating based on the area the plant actually takes up, even though you planted on 6 feet, that kind of feels like cheating.
No, we're calculating both. That's what I was saying.
We did calculate both.
Both. Yeah, I know it's both, but for the efficiency part,
we had
6
different
feet, right? Or just
conclusions depending on how we did it.
But it seems like it's cheating just to do it on the 4-foot spacing, 4 feet, because the actual is 6 feet, and you don't know if it was actually 4 feet, it would be more crowded, it might be smaller. So I don't know. That's just the devil's advocate.
I kind of wish I had presented the data on the plant size.
That's more of a way, it's not, that measure we don't really use as a production thing except for extrapolating, like it wouldn't factor into like the calculation that Lois does at the end here for like what's the plant justified cost. We were doing that partly to see also just like how are these plants performing overall, because they're quite small. And part of the question we have is why, and the reason is mostly they've just been bearing pretty heavily for a long time, so that's kept them at that size.
But the other,
It's multiple measures that, like, not all of them go to the same question. Like what you're
Right.
saying is, that's where the lineal foot measure, which is like, no matter the size of this plant after this many years, it gets 16 for the, like, calculated yield per acre. Okay.
another factor there was, we had 2 years of drought in the formidable time of those plants growing. So they were probably held back between a combination of the heavy grass and the lack of water. So that could figure in on this.
Those weed mats though are very, very effective at conserving moisture. And so of all the sites to be affected by drought, I was less concerned about that. I wanted to end by saying that Mike Lilja's system of planting asparagus in the alleys was really a very wise way of getting economic value from that land while waiting for the plants to fill in.
I'm gonna fly through, hopefully, this update on clonal propagation, which is mostly a retread of what I presented last year, but there's some new results in it. Okay, so clonal propagation, why do we do it? Why do we make plants this way? Unlike grain crops, unlike most crops, woody orchard crops are planted with clones. They are vegetatively propagated. They're not grown from seed because you can't, over a long enough period of time, maybe you could, but in general, seedlings of woody plants are too inconsistent. So vegetatively propagated clones are how you get uniform yield, uniform qualities, uniform machinability traits, size, bush width, even ripening, harvest timing, harvest detachment forces, things like that. So it's how you get uniformity. Basics of what clonal propagation is you take some chunk of a stem of a plant that you like and want to clone, and you force it to grow roots somehow. And then you have taken little bits of a plant and turn each one into its own clone, its own individual one. Depending on the technique, if you have one mother plant that you want to clone, you can end up with dozens or hundreds, or if tissue culture works, thousands. This is a diagram of stem cuttings that I took off of Google, just a very simple, take a piece of a plant, force it to grow roots, now it becomes its own plant. Clonal propagation has been, probably still is, our biggest challenge as a program overall. It's been a lot easier to find good plants than it has been to be able to multiply them in large numbers. So it's our primary limitation to commercializing the varieties that Emily mentioned earlier that have come out of the breeding program. Lots of types of woody plants that clone very easily. Hazelnuts are not one of them. And you need lots and lots and lots of plants. 3,000 acres is over a million clones. There have been 4 or 5 of our varieties that are both agronomically desirable and have demonstrated propagation potential before. We have other varieties that have not been, that are anywhere from sort of mediocre to bad in terms of their ability to propagate. And there's a huge genetic component of it. Our breeding program has the full distribution of propagation ability. And then there's also another selection, Northern Blaze, from Cremo Nut Nursery, which is a private company that is also being commercialized, that has been one of the easier ones to propagate. So for a technique, you need to be able to make self-rooted plants, so not grafted like a lot of fruit trees, like apples. It needs to work. It needs to be able to go from one mother plant identified in a breeding plot somewhere around year 8 or in a farmer's field to making lots and lots and lots and lots of clones of those fast. It needs to be cheap, relatively cheap, because of the number of plants that need to be planted per acre for this production system, which is in general denser than a lot of other orchard crops. 400 plants per acre. They can't cost $20 a piece. So fast, cheap, easy, successful. So there's a couple of options. There's tissue culture. I'm not going to provide an update on that. There's ongoing work at UW-Madison. There are hazelnut varieties that work very well in tissue culture. Up to this point, ours have not been that way, and that's been something that's been true for more than a decade, but there is slow progress going on at UW. The other way, this is old reliable, which is mound layering, which is, these are quick diagrams from Lois. You have a bush that you like, you cut it down to the ground in the wintertime, new shoots grow up from the base, you pile a bunch of sawdust, put rooting around the base of the plants, you put rooting hormone on it, you can apply girdles, like twist ties or something like that around the stem. They grow roots into the sawdust, and then you come out and cut them out. This is the last. Yeah, you come in at the end of the season, and you cut them out of the sawdust. You've taken one plant, depends on how many shoots it makes, and you've made a dozen or 2 dozen or 50 copies or something of it, something like that. That's what it looks like at the research scale. We usually do this on the hottest day of the year. That's the time, it's time to start mound layering. Everyone's invited. So this is thinning the plants and putting on the twist ties, applying the rooting hormone. You can do this more efficiently than we do it, but this is how it works for us. This is what the plants look like after all the sawdust has been applied. So these are starting to grow and root into the sawdust. And when it works, this is variety State N76, which we're calling Trail Runner.
Yes.
This is one that propagates beautifully. You can see at this point we cut them out of the mounds with a sawzall, and when it works, they're so rooted together that it just sort of comes out as a big mass like that. And that's what the individual plants look like. That's a nicely rooted 1-year mound layer, and you can plant that right in the field. So we do it the silly way, laying down on our stomachs in the field, but this is a mechanized large scale process. This is used for almost all of the rootstock industries in the world. Apple rootstock, fruit tree rootstocks are mostly done by layering, and the world hazelnut industry is mostly plants are produced that way. And then third option, which I had a diagram of before, is stem cuttings, which is take a chunk of a plant, stick it in a pot, put it in a mist, or apply rooting hormone, put it in a mist house, force it to grow roots, and get an individual plant out of it. And they have the two techniques between cuttings and layering have advantages and disadvantages. Cuttings are higher multiplication rate. You get more plants faster. You can produce plants that somebody can plant in the field, leaf on or dormant, which has itself has advantages and disadvantages. The downside of cuttings is that mist houses are, an expense, and there is no tolerance for failure. You basically have these plants that don't have any roots. They're on life support for a couple of weeks until they can force out roots and grow on their own. So it takes a lot of expertise and a lot of skill to manage them. And layering is just sort of the other side. It's sort of old reliable. It works for the varieties that it works for. It doesn't have as high of a multiplication rate. You can only end up with bare root dormant plants, which a lot of growers prefer, but some might not. And at this point, we don't really know where the breakdown might be commercially between what can make a cheaper or better, or however you define it, plant, stem cuttings or layering. And I should say, cuttings until the last couple of years have really not worked well, but that's something that has changed, which is great. So we need to scale this up. We need to work with professionals. So the UMHDI has relationships with Curtin's Nursery and Hauser's Superior View Farm. That's also a nursery that's up in Bayfield. Curtin's is local in Inver Grove Heights. Curtin's is primarily focused on commercial layering at this point, which they're scaling up for, and Hauser's has been working with cuttings and did a really great job this year. This is much better than we've seen before. So this is a graph of, the different varieties, their success rates, large numbers, and this is still with a lot left to optimize in the process. But this year we were planting in mid-September plants that looked like that were, cuttings about this big, 6, 8, 10 inches, something like that, harvested in the field in June. And we planted, these are, so this is the first, this is a first for us, having plants produced that quickly, that efficiently. And we field planted about 750 of them on a couple of different go-first farms, these initial stage commercialization farms. And I believe that those are the first plantings of plants produced by commercial nurseries going onto this farm, as opposed to mostly ones that we've been generating in-house at the university. Hauser's numbers were really great this year. This is like an order of magnitude beyond what we've done before, but a lot to improve. You know, over 5,000 successful ones from around something like 6,000 cuttings stuck. And we're excited about it. Gertens is in the process of planting out their site in Denmark Township, commercial scale stool beds from stock plants that they've generated themselves or they've been transplanted from university plants. That's where we're at. So yeah, this was an encouraging year. There was a lot of progress made, and there's a lot more to go to getting plant price down, really making the success rates consistent. Yeah, getting this to viability. So that's where we're at.
We had a question online from Patrick Young. Does air layering work with hazelnuts, and if not, why?
I haven't tried it. I've only heard horror stories about it.
Air layering is pretty much something that's mostly useful for houseplants. I once tried it in the field, and what I found was the wind just decimated it.
What did Stowhouse or what did they do differently?
Hausers?
Yeah, Hauser.
They're really Dane Hauser, who manages it. He's got some tricks up his sleeve, but he's in there like all day, every day. And I think the management has just been a huge part of it. And they're able to do things that us as like university researchers or whoever, or student workers running around like with a bunch of other things going on, haven't managed to do. And he's been propagating, plants out of mist houses for like 10 years or so. So he really knows what he's doing. His system is dialed in. So.
But also, there are two other things to his advantage. One is that he's primarily a bedding plant producer, but by the time the hazelnuts come through, the bedding plants are gone. So it's just hazelnuts. So he's only managing hazelnuts, not trying to juggle them with other things. The other advantage that he has is that, at least this is the theory from our colleague Jason Fishbach, he's up on the North Shore, he's at the north end of Wisconsin, just south of Lake Superior, on a north-facing slope. And so the breeze comes off Lake Superior. So he doesn't deal with the massive fluctuation in greenhouse temperature that greenhouse managers in other locations have to deal with. So Jason lives in Ashland, which is, maybe 20 miles away, further inland. And Jason has some hoop houses, and Jason says that there's no way he could do what Hausers does.
That's a compelling reason.
Yeah.
There's a lot of nurseries that the Great Lakes.
Yeah.
Partly for that reason.
Yeah.
In terms of making it work for other nurseries, which I think is critical, I'm increasingly coming to the conclusion that the research has to occur at that nursery under the conditions and facilities of that nursery, that they're going to be highly variable from nursery to nursery.
Mm-hmm.
One problem is he's not been producing his own cuttings. Mark has been delivering most of it.
And that's something we need to fix. Yeah.
At this point, a lot of the material is still from plants that are on university plots. So June is a big plant shuffle, but I prefer it to mound layering, which is how we usually spend a lot of June. So
You got stock plants too, or what's the situation with that?
So Gertens is the large retailer and wholesaler for 9 states. I think they take truckloads of bare root, truckloads of potted carts worth to other smaller nurseries all over the upper Midwest. And their growing ranges dedicated for woody tree propagation is pretty substantial. They've got about, I think now 12 acres under greenhouse glass with heated floors, LED light controls, heavy on the greenhouse management for integrated pest management. And they've been doing this for almost 100 years. So they've got capacity. 5 years ago, when I started working with Lois in the field, they didn't know much at all about hazelnuts, but we've had several cycles now for them to increase their learning curve. And in a nutshell here, I'll just give you last year and the direction for this year, and we can talk about specifics as we get through this. So last year, we had transferred the seedlings for the Gophers Farms that you had provided the seed net for. There were 1,600 potted plants. The potting part of it versus the in-ground is one of the biggest differences in how these plants function in maintaining them, trying to get them to grow, trying to get them to be responsive for different stages in their life. They're very finicky. They do not like transition at all. You miss 2 or 3 watering cycles, they start to argue. You put them on floors that are too warm or ventilation that is too fast. Heavy wind will desiccate them. They are very, very tricky in the pot. You put them in the ground, they get nice and settled. And that kind of comes to bear with what everyone is finding with mound layering as a technique to clone and propagate these. Gertz was working on their first grant that ended last year, and now they're working into their second grant. The second grant is budgeting for a scaled 2.5 acre, as Mark mentioned, mound bed layer, just like what we're envisioning for his cherry orchard image. Had heavy mounds on the bottom of every one, but they were long rows. Gertz now has access to a custom harvester, which basically cuts through all of those new roots in a mechanized fashion. So there's no laying on the ground trying to propagate them. You build them up as a shoot mass, cover them the same way, but not twist tying them. So the labor gets reduced substantially. And then you cut out below the new roots and above the existing crown. And you can do that repeatedly every year because those shoots are in fact feeding the stable standard in-ground mother plant roots. And so when you reduce the top part out, you're not affecting the root base, including continuing taking cuttings. There's even supported in the grant, there's even a tissue coating or a tissue culture trials. And in Oregon, there are very successful nurseries that are initiated with tissue culture plants, but they grow them out directly into the mound bed environment. So there's a lot less involvement with potting and overhead in the nursery. None of the infrastructure, none of the overhead goes into that cost of that plant. It is simply propagated to some stable size and placed in the outdoor soil and then managed for mechanical harvest. That is the model that Gertz is putting 90% of their effort behind. If we can get tissue culture plants at scale from wherever and cuttings at scale from wherever, all of those at the Gertz strategy is to put them in these mound bed layers and bring out not just a 3-year-old plant from a pot to go into the field at Gophers Farm, but an actual substantial rooted bare root plant. It's going to be much more effective for stability in its first year for dieback percentages from the smaller plants versus the larger plant. So we're trying to build that into the strategy for production also. Gertz has talked with the University of Wisconsin in the building of this current grant on a tissue culture basis, and we couldn't get it to work. They wanted to have the grant pay up front for royalty fees, even though they did not have product that they would guarantee or protocols that were yet proven. They still wanted the royalties up front, and so we decided that wasn't the right direction for that money. And so there are tissue culture trials going to be started with sterile tissue here at the University of Minnesota. The grant allows for education, allows for production, allows for tissue culture per plant costs to be able to be generated and transferred to Gertz to be able to grow them out. So in some ways, we are trying to piggyback on all of the recent research and propagation findings. And after talking with Jason and Amaya in Wisconsin, they're scaling up theirs also, their program also. So we would, according to the last conversation and by working through the support letter for Amaya, hers would be one of very few nurseries to receive plant material, tissue culture plant material from them to be able to grow out at scale. So we're pretty excited about that.
Yeah, well, you've got some nursery, you've got some stock plants now. Where are we in terms of starting actually producing plants from those?
So we have, there are, I would call it 10,000 stems that I have right now. There are 35 plants, mother plants with large established crowns at one of the growing ranges that was toppest last year in late April. They've grown up substantial potential stem cuttings or node sections, and there are right now plans to do hardwood cuttings to reset that plant. One, to try to propagate the hardwood cuttings, rooting them with dormant buds, and the other is that once you coppice them, they will immediately flesh up another 5,000 to 10,000 shoot green shoots, which then can be harvested for green tissue rooting. And so there's a kind of a double harvest this year. But eventually all of those mother plants will be transferred from one location where they were stabilized when we first transplanted them to the dedicated 2.5 acres out by Afton State Park.
You mentioned the 4 or 5 varieties that have names.
Yeah.
And are being put out there. Are any of those the plants that are being produced in these propagation programs and the layering? Is that the set you're working with right now?
The reason why I'd like to go back to Mark's table, which showed the different varieties. So you see that Rose 14-12 is at the top, or actually you see that there are 5 varieties that are shaded. Those are our top ones that have that we're releasing. Mm-hmm. So the reason why we are releasing those ones is because we've had relatively good success in propagating them. That was based not just on stem cuttings, because those decisions were made before we had the success with stem cuttings. They're based also on layering success. So for example, Stape N7-6, which we call Trail Runner, which shows only 53% success with stem cuttings. That one actually roots phenomenally well in layering.
Yeah, so there's...
And it produces a lot of suckers. Let me finish my answer and then you can say what you're gonna say. So basically, the reason why we have chosen those plants is because they do well in layering. There are other plants that have just as good or maybe even better agronomic characteristics in terms of yield and nut quality that we haven't been able to propagate successfully. That Rose 14-12, we propagated it even better. I don't even remember what the agronomic characteristics are of it because it's been so long since I've looked at it.
Yeah, there's some fluky aspect. I'm glad I sort of glossed over this because I was worried about time and I also forgot. There's some interesting sort of fluky things with this slide. So like the variety that Lois was talking about, Stape N7-6. That's been like consistently along with Rose 9-2, like consistently head and shoulders above the other varieties. And like last year in the same system, it's like at Hauser's, it was like 88% with a much larger number. So it might have something to do with just like the particular quality of these cuttings. One of the things that's been nice is that a number of the varieties that are shaded, that aren't shaded, that are in white, are ones that have been kind of like mediocre or hit or miss in mound layering. And we've done them for enough years, like Lois was saying, that's like our benchmark for its like kind of genetic ability to propagate. But there's a number of those that are ones, so like Art 4-3, Gibbs 5-15, Eric 4-21. Those are ones that have been mediocre to like almost impossible, like just really bad at mound layering, like nothing that was seen commercial. This year Hauser's was having, really good or really encouraging success with a number of them. with large numbers. So what we're hoping is that there are some more varieties that are sort of brought to the fore that that weren't looking good from just straight mount layering in past years.
And two of those varieties that are shaded as being ones that we're releasing, State N7-6 Trail Runner is the new name, and ARB 7-1, which is Adventure. Part of the reason they did so well in layering is because they produce a lot of suckers. So it wasn't so much a percentage rooting factor as just we started with so many. And State Bend 7-6, in fact, we kind of started pulling back on propagating it because we've got so many of them. We want to keep more balance what's out there. I also want to mention, I didn't present any data on our new generation of material or our joint performance trials that compare these side by side together with material from other places. I haven't finished the data analysis for those yet. But with our new generation, which are crosses with Oregon, selected from them, so far in layering, we found that they, in general, as a group, not entirely, but as a group, they propagate a lot better than this material. And the explanation for that could be simply that they are descended from plants that propagated well, because the Oregon industry was, and all commercial industries were selecting plants based on propagability as well. Whereas our mostly wild stuff, you know, nature doesn't care about whether you can layer them. So what that probably means is that as we develop newer generations, the propagation bottleneck will not be as big of a problem.
There was one other factoid from this that I forgot to put in. So we had, and this is sort of like a layering versus cuttings thing. From one site where we propagated cuttings of R7-1, we had the past couple of years of mound layering those same exact plants with, you know, number of stems that were layered, what the success rate, what the rooting rate was. From just one date of harvesting cuttings out of 4 of those varieties, we harvested slightly more cuttings than total number of stems layered on those same plants, and that the overall rooting rate of those stems in mound layering the past year was somewhere around 50%. It depends if you count the really, really weak ones. The success rate of the cuttings on that date was 92%. And then we had 2 more collections that were about the same number of cuttings off of those same plants. So that's one of the things that cuttings really has going for it, if you can force it to work, is that the numbers are, you know, double, triple, 6 times, at least for one of the varieties.
Another thing to put in the advantage of cutting column. Layering is really a lot harder on the mother plant. So we see that the first year we layer a plant, we have fairly good success. The next year we have lower success. The third year, we probably should skip it just because it's so stressful on the mother plant. And the reason for that, the method that we've been using for cutting, for layering, involves putting these girdles around the stems, which means that you got the stem going up. It's photosynthesizing under normal conditions in nature. It would be sending photosynthate down to the root system and nurturing the root system. That girdle blocks that translocation, and so it's basically starving the root system. Now the way around it is to leave some stems that you don't layer, but if you don't leave enough of them, and those stems also end up shading out your layers, so eventually you're gonna kill the mother plant if you overdo it. Whereas what I've observed with stem cutting, so far we haven't been doing it that many years, but we've been doing it enough years that I think it's true that you could probably do it year after year after year. Because once you collect the cuttings in early summer, you might collect them 3 times, then you let them grow up. And right now, the plants that we did this to last year, they look like normal plants. I mean, normal plants that were coppiced in that spring. So they might have stems that are this big. We'll have to coppice them again this spring, but they'll do it again, and they'll do it again, as long as we keep on fertilizing very well.